Okay, so I think most of us has joined. We can see 15 participants here. So first, I would discuss about the progress of the course till date. So for that, we will be going to this link. This is the organization link of the educational platform we have, edu.t-bio.info. Now let us go to the educational place and then try to figure out what are all there. So this is the place where you can go to find out about few things about the progress of the course. So there is a home tab here and you can find what about the upcoming program. The today's program tells you about which is 5th September. It tells you about the program itself and also the details that are going to be covered in the program. And besides that, there is something called forum. The forum tells you about, let us click on the forum tab and try to figure out what all are there. The forum tells you about all the files that needs to be at your desktop for the presentation to follow, and also the presentations and the files of the previous workshops. And at the same time, you can post your questions here. You can post about your any doubts that you have regarding the session, regarding any questions you have in your mind. You can give it a title and you can write about your question here, and then we will be answering that accordingly. So the forum is meant for an exchange. So the exchange of information, the exchange of data, the exchange of knowledge. So that is why we are trying to build a forum of commonly interested and initiated participants who wants to learn about big data bioinformatics and wants to learn about the data science perspectives of biology. Besides that, there is a button called progress. About the progress button tells us about how the course is going. Where are we right now? Let us see where are we right now. How much you have been able to go forward. So we also can find several people who have joined the program. This is specifically meant for us. We at the administrative side, we know about your progress and we can guide you in specific process so that you can have a better outcome about at the end of the program. So let us look at the progress so far. See, there is a lot of activity I can see on the server itself. Probably you people ran a few demo pipelines. And here is a leaderboard. The leaderboard tells us about how many people, what are the people doing. Shubhrajit has been working on several projects right now. So he is using a lot of server. That is why he has got points on the server itself. And Devalina has been using a lot of educational. She's going to the education portal, trying to finish many of the courses, I guess. Arpita doing the same thing. Ruchika. So one thing I would like to ask you is, if you go to your profile, for example, if you click on this profile, so she has a photo definitely, but what needs to be done is to improve your profile by giving yourself a name. So the name by default which is taken is the name of your email ID. So if you are trying to get yourself identified, this is good to have a photo. Thanks for that, Ruchika. And at the same time, you need to put your name properly here so that we can identify you by the name itself. So that is the thing you should be doing before the next class probably. For example, she's making good progress. As you can see, she has passed the course Introduction to Bioinformatics and so on and so forth. So, and she has collected points, 600 points. So she's at the beginner level. This is the batch you can see at your end, and the batch would tell you that how much progress you have made. So you can evaluate yourself at the same time to become from a beginner level to a mid-senior level to a senior level and to an advanced level. So that is the importance of the forum. And if we go back to the organization page, there is something called curriculum, assignments, and courses. But at your end, you probably would be able to see only home, forum, and curriculum, because the rest of the tabs are meant for the course administrators like us, and also the course administrators at the end of the university. So this is a very interesting view of the entire length of the course. You can scroll down and figure out that what all sessions are going to come up and what all are to be covered in the session. So we have passed all these steps. Now we are at the level of Transcriptomics 2, and then the next session is going to take place on on September 10th, which is a workshop again. So I will talk about that later on, specifically for the SNU students who have joined. So let us go back to the presentation. So this is all about the organization page. Review about whatever we have covered so far. We have covered in the last lecture, we have covered about processing NGS data, transcriptomics data, RNA-seq data. We have used pipelines, demo pipelines in our server. We have learned about the role of pre-processing in the standard RNA-seq pipelines. We have used algorithms like Trimomatic and PCR clean. We have tried to understand why these are important in the pre-processing of the data as a part of the pre-processing step. And thereafter, we did some mapping onto the genome, mapping onto the transcriptome, and also we used something called combined strategy, which tells us about how the things go around. So a little bit of overview of the things that we did. We did pre-processing, basically 2 steps: adapter removal plus additional trimming, removing the PCR duplicates, followed by mapping onto a set of known transcripts and mapping onto the genome and potential identification of novel transcripts as well. And at the same time, we use something called combined strategy. All these steps of pre-processing and mapping, after these steps are over, we come to a point which is connected with quantification of the gene expression. As a part of this, as I was telling you, we use 2 pipelines. One is this, where we have started with PCR clean and Trimomatic, and thereafter we directly mapped the entire results onto the transcriptome by using an algorithm called Bowtie2T. And thereafter, we generated a table of expressions by the algorithm RSEM table. And this is also a demo pipeline that we have used. And I guess that this was, you people have run demo pipeline. The sole purpose of setting up the demo pipeline is to make your life easy to understand about how to, what kind of input files are required for your run and what kind of output files would you expect. And the most important part of setting up the demo pipeline, running it properly, is that typically demo pipelines does not require much time to finish, and within a short span of time you will get to know that what can you expect out of the pipeline itself. Another pipeline we ran is this is a combined strategy pipeline that we ran. We started with PCR clean and Trimomatic. Thereafter, we use an algorithm called HiSat2. HiSat2 is specifically for mapping onto the genome and also mapping onto the junction regions. We understood bits and pieces about what is the importance of junction, what kind of biological significance does it contain, and thereafter we built the isoform construction using cuff links and cuff merge. And also at the same time, after that, the usual step of mapping onto the transcriptome, which is by bow tie 2T, and generating the table of expressions by RSEM table of expression. Okay, and another RNA-seq overview that we are going to kind of touched upon is starting with preprocessing, mapping onto the genome and known transcripts, thereafter quantification of the expression levels, and differential gene expression. Now, a differential gene expression is something which we would like to look at a little in a bit more detail today. The pipeline that is there as a part of the demo in the platform itself is this. Now, in addition to whatever steps that we have used so far, starting with preprocessing like PCR clean and Trimomatic, thereby going to HiSat2 and cuff links and cuff merge, bow tie 2T and RSEM table of expressions, combined strategy, and we use an algorithm called DESeq2 for getting the differential gene expression. The file that would be available to you if you use the demo pipeline are these set of files. The output of this extended pipeline that you saw in the previous slide is a series of files, among which we would be interested to look at in the highlighted file that you can see within the square box. Now, what does this file contain? We get the filtered gene expression table in FPKM units. You possibly remember that I touched upon this topic of FPKM. Now, to reiterate the fact, at FPKM values are frequency per kilobase per million. And we also discussed about some other thing called RPKM. And at the same time, we also discussed about TPM, transcripts per million, and so on. And in today's session, we will learn about the analysis, next level of analysis. So about it, little bit of details about the file that you can see in front of your screen right now. The column that you can see highlighted is the sample name. The name of the sample is right there. And what else we have in this file? We have a gene ID, which is an ensemble ID, and we have the level of expressions. What are these expressions? These expressions are FPKM and TPM, as I was telling you about. Today we start with something called Transcriptomics 2, September 5th. And what we are going to cover in today's session, the first step at this level of analysis is quantile normalization. We will also learn about principal component analysis, which the basic idea about principal component analysis is reduction of the dimensionality of the data and separation of the data into groups. And whether this group separation has occurred properly or not, we would try to visualize that data. We would like to see that, how does it actually look in separated form. And then we will look at something called factor regression. Factor regression happens to be an advanced machine learning algorithm. We start with normalization of the sample data. Normalization and preprocessing are essential steps for the analysis of high throughput data, including next generation sequencing and microarrays. Multi-sample global normalization methods such as quantile normalization have been successfully used to remove technical variation from noisy data. These methods rely on assumption that observed global changes across samples. Mohit, or anybody else? Could you mute your microphone, please? So I would request you, anybody who is joining a little late, I would request you to mute your microphone. This session will be recorded, so it will be a little disturbing for you guys when you go back and listen to the recording itself to learn about the stuff. Thank you very much. So I was talking about that multi-sample global normalization methods such as quantile normalization, have been successfully used to remove technical variation from noisy data. Now, these methods rely on the assumption that observed global changes across samples are due to unwanted technical variability. Transforming the data removes these differences, and this has the potential to remove interesting biologically driven global variation and therefore may not be appropriate depending on the type and the source of the variation. Quantile normalization, to be very specific, is a global adjustment method that assumes the statistical distribution of each sample is the same. A very interesting example that I came across recently, it said that it was observed that some of the data sets formed by quantile normalization combination in some inter-study validation research had superior prediction performance compared to the data sets combined by other methods. Now, combining data sets using QN or quantile normalization could improve the prediction performance for that study. Things become little interesting from the next slide. On the left, you can see these are the raw signals. Signal coming from raw FPKM values from samples, as you can see, are clustered towards the origin, while the outliers, which are more visible and pronounced, represented by the distributed dots, and the red line here is the regression line. Regression line is something where we would try to fit our data so that it become a standard normal distribution. Because raw reads expression is not the correct amount of data, correct type of data that can be used for any sort of data science principles or statistical principles to be used. So upon transforming them into normal logarithm with base 10 or base 2, the values become more even and normally distributed, as you can see. The meaningful values become more prominent. The outliers would already be there, but as you can see, the chief cluster is right here, whereas on the left, the majority of the values, which are standard normal distributed values, are clustered towards the origin. So during the process of normalization, the input parameters, you will be using these quantile normalization processes in the server itself. So during the process of normalization, the input parameters that would allow you to log normalize the data. Now, in raw data, another representation. So representation data visualization is a very important part of descriptive statistics and also inferential statistics. So each sample has been plotted here, as you can see on the left. These are the normal-like samples of breast cancer, and these are the Claudin-Low samples of the breast cancer. In raw data, in each sample in normal-like or Claudin-Low subtypes of breast cancer, before normalization the significant values are invisible, and also the mean, sum, and the sample variance also depicts the same story. So see, the difference is pretty significant, and this is significant but not statistically significant. Now here I would like to say that in addition to quantile normalization, there is another normalization called rank normalizations, and these two methods are widely used as a part of pre-processing technique, and these are designed to remove technical noise presented in the transcriptomic or the genomic data. Subsequent statistical analysis, such as gene differential expression analysis, is usually based on normalized expressions. Upon normalization, as you can see, the distribution is more symmetric for two subtypes of breast cancer. And now statistical methods can be used on them. There's another important concept that we come across right here is kurtosis. What is kurtosis? Kurtosis It is a statistical method or a statistical tool or a statistical feature, I would say, that is used to measure extreme values in either tail as in both ends, these are the tails of a graph. And either tail, and also describes the data around the average. Distributions with high kurtosis exhibit tail data exceeding the tails of the normal distribution. Distributions with low kurtosis exhibit tail data that are generally less extreme than the tails of the normal distributions. So after log transforming the data, the distribution becomes more even. And now again, to reiterate the fact that appropriate statistical methods can be used to bring out some insights. The method of quantile normalization is also associated with some selection of threshold value. With threshold zero, the process fails to capture the variance within the data. So first log normalization and then quantile normalization. The basic purpose of it is to remove the noise and to bring out the signals. So thereafter, we do something like this. Hence, we select a threshold value of 5, which allows us to capture the normal gene expressions and also the significant signals as well, and at the same time removes the technical noise. One other reason is that quantile normalization ensures that the median values are captured. Quantile normalization is a technique to define it in a proper way. It's a technique of mapping original values to the corresponding quantile of a standard distribution. So few terms you have to remember very clearly: standard normal distribution, kurtosis, quantile normalization, and so on and so forth. So you will encounter a little bit of interesting features here and a little bit of interesting statistical keywords which would come to your use when you will be doing your project, or you'll be doing reading about them in the scientific papers. Here, an estimation of the sequencing depth of an experiment is determined by the upper quartile of its count. Okay, and comparing quartile values instead of raw values will eliminate differences by which are solely rooted to the technical variation, such as amount of RNA sequenced, such as sequencing machine type, or even unexplained differences that arise between different technicians following the same protocol. So when a particular technician goes into the lab and try to analyze or segregate the RNA data from the cells, fishes out the data from the cells. So the amount of RNA, the sequencing machine type, and we have studied a little bit of about the sequencing machine types. We would study more about in the next few classes. And also different technicians have different expertise level, different skill sets. But although they follow the same protocol, these kind of mechanical artifacts are obviously included into the data. And before going into the next level of any analysis, we need to remove them. So that is why transforming them into the log scale and quantile normalization procedures as a part of pre-processing plays a huge role. Okay, so going forward, when a set of approximately normal data is visualized via a histogram, it shows a bell peak. It shows a bell peak. It's a bell curve. This is typically called a bell curve or a standard normal distribution. And most of the data is falling within plus or minus 3 standard deviations from the mean. However, when high kurtosis is present, the tails extend farther, and thereby it is more than plus or minus 3 standard deviations of the normal bell curve distribution. So these all important features you have to take into account when you are trying to analyze your own data as a part of a project which you are going to do as a part of this course, and at the same time when you go back to your research and try to analyze the big data out there. So all these processes are called descriptive statistics. Here is the summary of whatever we have shown you. The series of bar plots in the above figure summarizes the finding as a result of log normalization. You can see that after log normalization, the data has become more even, more normal, more symmetric. And this is the descriptive statistics series of bar plots that you can see on the screen. It summarizes the findings as a result of subsequent quantile normalization with a threshold of 5. In addition to this, we also would like to draw box plots of our data. When a raw data is plotted in a box plot, the significant values are clustered together and are not visible at all. See, and all those outliers are visible on the top, and the significant values are all out here, while the outliers stand out. But after being log transformed, the mean and the median of the data come together. What does it really mean? When the mean and the median of a particular data set come close together, then we can say this is a symmetrical distribution, and thereby can be used proficiently for the next level of statistical analysis. Same thing for quantile normalization. Similar things, it goes for quantile normalization with a threshold value of 5. As you can see, the mean and median are almost at the same level, and thereafter we can use it for the next level of analysis. Now, what does the normalization procedure has on the replicates? The replicates are the values that we have that we have. These are the data sets. These are the data points. Applying the necessary methods of log transformations and quantile normalization, the replicates are more symmetrically distributed, as you can see from this graph. Okay. And what are these processes of normalization and log transformation have on differentially expressed genes? These definitely affects the differentially expressed genes. Now, raw reads have more noise, as you can see. The raw read have more noise and mechanical artifacts. Upon log normalization,
excuse me.
Yes,
can you go to back slide? Yeah.
Please speak.
What is the meaning of midline? Because I had read a paper and exactly there is a similar result, but I am not able to understand. Can you explain the result? How can we analyze like the in between the middle line and the yellow color and the blue? Because I am not a student of bioinformatics.
No problem, no problem. Jyoti, happy to help you.
Okay.
Happy to help you. So, could you just hold on to your question? Can we address that towards the end of this lecture? Can I go on?
Okay.
Could you remind me at the end?
Okay, no problem.
Yeah. So, but quickly I would tell you the line that you can see on out here is called the regression line. So any data point, when we try to do statistical analysis, we always try to fit the data along the, so this is the ideal situation. This is what should happen. But due to several processes of different processes of data generation, what happens is the data is all over the place. So we try to apply algorithms, we try to apply statistical measures, and thereafter we try to bring the values closer together, closer to the line, so that we can explain the result properly. So this is quantile normalized zero. So as I was telling you that in a few slides before, that there is something called threshold value, right? So threshold value is used when you are quantile normalizing it. We will show you in the platform itself. So log transformed versus quantile normalization with a threshold of 0. And this is the graph of log transform but versus quantile normalized 5 and the data expression. So comparison of the replicates. So when we compare the log transform versus QN0 versus log transform versus QN5, you can see the distribution is more even. The distribution that you can see, the blue colored dots, they are more So this is also called the linear or the log normal scale. This is also sometimes called, other name of it is regression line, but here in this case this we call log normal scale. So when we normalize the data, until and unless you normalize the data, you cannot do any sort of statistical analysis with the data itself, because raw FPKM values, raw read values from the RNA-seq experiment does not follow a standard normal distribution. It has to be normalized first. Then the next level of any sort of statistical analysis can be used. Does that answer your question, Jyoti?
Yeah. Thank you.
Okay, no problem. So get back to us. If you have any more doubts, we'll address it separately.
Okay.
Okay. Thank you, Jyoti, for asking the question. So yes, I think Jyoti has a very good, very interesting question to ask. She being a biologist, and thereafter these courses that we are doing are typically meant for biologists who wants to do bioinformatics at their end, who wants to do increase their bioinformatics skill sets, and these courses are entirely meant for that, so that we can explain to the core biologists that what kind of data analysis skills they can take up or they can acquire to make the data that they are producing more meaningful in terms of publication, in terms of good scientific research. So as I was telling you, the quantile normalization procedure and the log normalization, what does it, what effect does it have upon the differentially expressed genes? So these are the raw reads. As you can see, the differential expressed genes found by this procedure is 194, which is abnormally high. When we apply log normalization to the data itself, the number comes down somewhat to the level of 133. But when we apply quantile normalization in addition to the log normalization, it comes down drastically to 32. And remember that this is done at the threshold level of 0. But when we quantile normalize it in at the level of 5, then it comes down. This is a significant number of, these are the true signals within the data set, which you might like to look at for your next level of analysis. And here is this graph specifically shows you of a particular gene, the gene in different samples. These are the samples that you can see. So, and the individual color lesions will tell you about the whether it is coming from the raw reads, when the gene has been found among the raw reads, when this particular gene has been log normalized, thereafter quantile normalized with a threshold of 0, and thereafter quantile normalized with a QN of 5. Okay, is there any question in the chat? Let me look at. Okay, let's see. There are a few questions in the chat as well. Okay, yes. So yeah. So no more questions, I guess, and I would go on. Okay, now we would go to the server, and we would try to understand what we would be using. A little bit of we'll be showing you the typical pipelines that we use for this sort of analysis. Now we have done our part with RNA chip-seq parallel analysis of NGS and microarray data. We use the normal the demo pipeline associated with it. And today we'll be talking about unsupervised analysis. As a part of that, we will be looking at some PCA pipeline, and also the PCA is also there as a part of utilities and factor regression or factor analysis. Now let us go to the server and find out. Okay, and here we are. So all of you have probably have the login credentials to the server itself. Now, if you have logged on to the server, you would be able to see something like this, that you will be seeing your login ID, and you can see all these things: profile details, pipeline management, reporting, and logout. So this is the server interface that it looks like, a consistent interface, and which are point and click interface, we also call it that way. So we used the demo pipeline out here, and we have run it. So we can also, I can quickly show you the extended pipeline that I kind of showed you in a brief encounter. So let us go quickly and find out. Tremomatic followed by PCR clean, and then we click on the combined strategy of using HiSat2. So these are all educational pop-ups that you can find and you can read about it. It also has publications associated with it. So you can also go to the publication directly and figure out that, find out on your own that this is the reading material that you would like to ponder upon when you are at your, after the lecture or so. So thereafter we do our couplings. And please remember that if you keep your server open or, for a long time, it might be so that it becomes unresponsive. The easiest solution of that for that is to refresh the server, is to refresh it by the reloading the button, by reload page button.
Excuse me, Professor Sunita here.
Yes, Sunita.
Yeah. So I was.
But you are not audible properly. Can you
hear me now?
More clear?
Can you hear me now?
Yes, much better.
Okay, thank you. I was trying to download my own data using the GEO SRR files. Out of 12 samples, I thought of downloading only 4 or 5, but out of them, 3, one of them did not get downloaded because it said that there is no space.
Okay.
So what's the solution for that?
Okay, so could you repeat your question? So you could download only 3 samples. The last one was not, you are not being able to download?
Yeah, because it said the space is not enough.
Sorry, what did he say?
Space issues. It does not have, I think my more than 3 samples it did not download, saying that the sample size probably is more and the space is less.
Yeah, so that may be an issue, Sunita, because the sample sizes are typically in terabytes or something like that. So maybe because I guess these are all FASTQ files.
Yeah.
Okay, so FASTQ files have typically very big size, so it might not be possible every time to download the data like that. So, but maybe we can have you are sure that this data is free for public? There are many data in the NCBI SRA, which are proprietary data. They don't allow the data to download. There are, some of them are definitely free, but some of them are definitely require special permission.
Yeah. They are free data.
So these are publicly available data. So maybe we need to have a look at it, Sunita, at some point of time. So
okay.
I guess that would be a good next step. Okay.
All right.
Okay.
Thank you.
Thank you, Sunita. So clicking on the next button and going on to Bowtie 2T, which addresses mapping onto the transcriptome. And again, these sort of publications we have put up out here for your greater understanding. And then we generate the RSEM table of expressions. In addition to that, we would do DESEC2. This is the file that, this is the algorithm that I was telling you about. And this would generate, we discussed some of these things in the last class, if you remember. And there's also a publication associated with which is genome biology. And you can copy the link and follow the link to read more about it. So be very sure that the lectures are meant to give you pointers. The lectures are meant to give you broad as well as little bit of detailed overview of the stuff that we have in the educational platform. But at the same time, it is imperative that you go back to the educational platform, follow the courses. That's why we have put up the courses out there, and we have organized them in such a way so that it becomes a good experience for you to learn about the stuff that we are telling you right away. So we end the pipeline, and here if we look at the output files, so if we look at the output files, these are the set of files that we get. We get isoforms, not filtered, isoforms, we get expression isoform, a stat table, all the results are in a zipped file, and we also get not filtered FPKM from the also all these files are generated from the DESEC algorithm that we have used. We would be interested to in our analysis we would be interested to look at the fpkm.txt file in a few moments from now. Okay, let us go back to the presentation. And the next interesting thing that I would like to tell you is principal component analysis. Now, principal component analysis, it is a unsupervised machine learning procedure, and this is a method for compressing a lot of data into our representation that captures the essence of the original data. Now, as I was telling you, this is specifically meant for reducing the dimension of the data, because any sort of experimental data or the type of data that you are trying to handle here, gene expression data are typically very huge and being multidimensional, so it has to be brought down to a dimension which is understandable by the human. So we know about the data that you can see, the graph that you can see right now in front of yourself is two-dimensional, the y-axis and the x-axis. The other axis, what you can add out here is z-axis. So it becomes a little more three-dimensional. But if the data has so much amount of dimension, it is difficult to really give some, have some insight from the data itself. So we will study more about PCA and principal component analysis to be specific in our machine learning course as we go on. But this is just to give you just to introduce you to the concept of principal component analysis. Now, this is what the principal component analysis pipeline looks like. Now, if I go to the server, I would like to show you how that thing occurs. So you can start right here. So instead of, there are two algorithms out here, visualization, PCA-R library. And here you can find, remember I was telling you that the differentially expressed genes have some influence on the principal components. So here the principal components you have to specify out there. So default of principal component is 3. You can increase more, but it is wise to keep it as a default level. Let's do it right now in the default level. And also if you want to transpose the data or you don't want to transpose the data, so it is better to keep it default and go along. And if you end, now it would ask you to upload the file. So let us name the pipeline right now. And you have to upload your file. So the file that you can upload out here, I have not provided you the file ID as yet. So you can upload something like, let's say this one. You can find this file in the Transcriptomics 1. So this is the file I would like to, sorry, not this is the file, this is not the file. Okay, so I will show you, let us do this way. I will show you a standard pipeline that I have already made. Let's not go into this. So let's say this one. So this is what is the file that this is the line, this is the pipeline that is being constructed here. So what does this pipeline tells you? So we have uploaded a cell line data, a cell line EXPR data. How does it look like? Let us see. Now let us save this file and look at it. Okay, so we have cell lines.expr data already, but again, we will, I could not find it that way. So my bad. So here I go. So let us look at this file. What does it contain? It's taking some time. So this is a tab comma separated value file. So it will be like that only. And if you click, it would open and will show you what this file contains. So this file contains the sample IDs. These are the sample IDs. Okay, and these are the genes. These are the gene names in the ensemble as ensemble IDs. So this is what is the resultant of the DESEG pipeline that I showed you. You can find this file from the demo pipeline, which this is the demo pipeline that let me go back. Sorry. So this is the demo pipeline I showed you a few minutes back. So this is the demo pipeline. From here, you will find a file like this. This is the file that I'm showing to you right now. Okay, and this is the file it would look like. So this is my input data into the PCA algorithm, and this will just imagine how many genes does it have. Let us look at it. So these have 6916 rows. So 6916 16 genes clustered into one place. Now if you really try to understand what is the differential gene expression from the set of file, it would be practically difficult for a human to do it. So that is why we rely upon the principal component analysis sort of algorithm, which helps us to identify the difference. Okay, so if you look at what is the resultant of this file, this will produce a plot like this, PCA plot.pdf. Now I have pasted the plot here in my presentations for your understanding. Let me quickly go to that slide and show you. So this is the generated plot that comes out of this PCA pipeline. Now, this PCA pipeline has an interesting feature. There is an R script which is generated. If you go to the server, you can find that there is an R script which is generated from here, right? So this R script you can download at your own machine when you learn sufficiently about PCA script, and then you can visualize it in your own way. We are in the process of setting up a course for specifically for biologists to understand about that, what type of visualization you can put, and these visualizations you can definitely use in your publication. So if you look at the graph is significantly divided into 4 quadrants, Besides that, there are many quadrants as well. Now these are the principal components. PC1 have 18%, so PC1 can explain 18% of our data, and PC2 can explain 17% of our data. So PC1 and PC2 can explain 25% of our data, and which is 1/4 of the entire size of the data that we are trying to visualize here. Now,
is it not Pratim 35%?
Yeah, I'm sorry. It is 35%. Yes.
Okay.
Thank you, Mohit, pointing out the fact. Yes. Yeah. Sorry. So wrong information again. Pardon me. So if you can find here that there is a distinct group separation between ER positive sample, estrogen receptor positive sample, and HER2 positive and triple negative breast cancer. So here, these group separation. Now this is known data. We know about it. This is the graph that we have done by a known set of data, trying to understand that how actually PCA works. Now, if it is unknown data, then this group separation will clearly tell you that what is the status of your data that you're going to handle. Now, this gives us a big insight of the data itself. Now, this is the fun of working with PCA, and we will cover more about PCA in our machine learning class, and you can also explore on your own as a part of the Transcriptomics 1 and also the Transcriptomics 2 course in the educational platform. Going forward, this is something I would quickly give you. We don't have much time left today. That's why I would like to tell you a bit about this, because this is very important and would come into use when you would be doing the project on your own. The variation that we saw in the previous plot between the two groups or conditions can be compared through a very straightforward statistical test or a t-test, which we have looked at in our previous lecture and also very covered in a lot of detail in the Transcriptomics 1 as well. But imagine when there are more than two variables that interact with each other and we need a more complex method of analysis. Now, factor regression is such a type of analysis, is such a type of a method that is often performed in experimental designs that include two or more factors, such as environment or time. Differential expression definitely is an useful analytic that we saw a few minutes back, but it has some major drawback. It only considers one factor. So that is why you can only compare samples along a single dimension. For example, if I give an example, it will be a little bit clear to you that you can use differential analysis to compare gene expression in heart disease patients versus men and women who never had heart disease. But you could not compare the influence of sex, whether which gender does the respondents belong to. If you wanted to look at differences according to the both heart disease status and sex, you would have to use a different approach. And one of those approach is factor regression, which you can see right now in the screen. Again, this is the regression line. So similar to the log normal line, log normal scale line that we saw in it. But again, here, the significance of the regression line is different than what you have seen earlier. Okay. Now, I have shared with you a file. This is the file which I have shared with you. This is a quantile normalized threshold 5 file that I have shared with you. Probably many of you have downloaded the file. Now we would use this file to do the factor regression. I will show you that how factor regression is actually done. Now it would be good that if you can follow with me, or you can just closely watch what I am doing, and you can replicate the entire process at your own end when the lecture is over, whichever suits your purpose. Okay, so I go to the server platform. I go to factor analysis. Here you can see factor analysis is completely separate. He's a lone soldier in the entire domain of algorithms that we have in the TBio platform. Again, it has an educational pop-up and tells you about the data formats that you can find out here, and it also tells you that what RNA seek and all this stuff. So these are all meant for people like Jyoti, people like all the biologists that we have out here, so that we can give you a guided experience, so that when you go back to your lab and do the experiments on your own, it would be very highly useful and highly helpful for you. So factor regression, and as we all know that this is an AI guided platform, the next immediate level is highlighted. This is some simple, very simple details about factor regression. It says that factor regression analysis is a regression based analysis of correlation between factors and patterns in the uploaded data. So you can find more details in. So here comes a place where you have to select groups. Now let us go back to the presentation and figure out what we have here. So as I was telling you, that you can compare between different groups, and each of these group can have individual components. Now here we are comparing between two types of PDX mouse models. So for factor regression analysis, we will use the result of the mapping and the quantification of the RNA-seq pipeline and the file that I have shared with you guys. We will select the samples from this table to work with the factor analysis, regression analysis. And we have two types of mice out here. One is skid node mice, and another one is an anthemic nude mice. So these are, xenografts model. So let me, I mean, I know all of you are, experienced biologists, you already know about all this stuff, but there are students out here as well. So that for them, I would like to quickly tell you about the fact. Now, skid node mice have severe combined immunodeficiency with no mature T or B cells. And anthemic nude mice lack the thymus gland and are unable to produce the T cells. Now, these are a part of patient-derived xenografts, and patient-derived xenografts, or PDX, are models of cancer where the tissue or cells from a patient's tumor are implanted into an immunodeficient, or they are also called humanized mouse. Now, PDX models are used to create an environment that allows for the natural growth of cancer, its monitoring and corresponding treatment evaluations for the original patients. Now, the significant common point between all these type of mice is that they lack host immunity. Now, what is this analysis going to show us? The different mouse models, such as SCID versus anthemic nude, might affect the gene expression of the tumor. Now, therefore, factor analysis with breast cancer subtype as one factor, breast cancer subtypes as one factor, and with the type of mouse, since the type of mouse affect the microenvironment. I just want to reiterate the entire thing. So factor analysis we will do with PDX mouse models as one factor, and also SCID and the mouse models here as the second factor. So we have 4 combinations out of the 2 groups. How will we do that? We will say a few minutes from now as I show you the demonstration in the server itself. So this analysis will tell us how does the gene expression among the cancer subtype change with the type of mouse, since the type of mouse affect the microenvironment of tumor and subsequently could have an impact upon the result. Now, this is the server pipeline that I've already run. I will show you how to do it. So how many members do we have in A? We have 2. Now, we want to add another group. How many do we have here? We have 2. Sorry. We have 2 here, and we have 2 here, right? Then we click on the next. Now we have to add file. The file that will be added will be the file that I have shared with you. So this is all right here. This is the file, PDX expression genes fpkm quantile normalized 5.txt. So let us upload that file. It says 100% upload, and then let's check. Now we have to divide. We have to divide according to the things out here. So we have ER node here. So let us do ER node in one group. So A1 and B1. And we have ER anthemic nude in the second group. We have triple negative and node in the third group. And we have triple negative anthemic nude in the fourth group. So now our data is ready to run. So factor analysis will try to find the correlation between the type of the PDX models and also the cancer subtypes we have under consideration. We do a continue, and now it will say that groups have been added. You can complete the pipeline. So see, for biologists, this is one simple step you need to take, and because you already understand the basic concepts of biology, we are giving you a platform which you can really point and click, but at the same time, it will also give you the understanding of how actual things happen. We do an end, and then you save. You have to give a name to the pipeline. Let's say 2 days, 2 days pipeline underscore 05092019, and then we save, and then we run the pipeline. So if you run the pipeline, it will be something like that, and this entire schematic diagram would come up. Now whether your pipeline has really run or not, you can find it from here, or you can go to the my pipeline and you can say, see, this is processing. Okay, so I have already run the pipeline, and I would show you the resultant of it. So this is the result that you can expect out of the pipeline. So let me go back to the presentation itself and show you. This is what we get back from the pipeline itself: the differential gene expression, the relationship between the different types of PDX models, and also the types of the subtypes of cancer for any particular gene. So the result that you get out of that, you can analyze it in such a form. So I would request you to go to the Transcriptomics 2 course in the educational platform. There's an elaboration of the entire process, whatever I am telling you, and if you can go through it properly, you will learn everything about how to run factor regression, how that would be useful. So the next slide tells you about that only. If you go to Transcriptomics 2, if you go on and do all the things and follow the the pattern that it has been organized into, you can find all the details out there. So technically I would end here, and before the question answer session, I would like to tell you about the next day is genomics and transcriptomics workshop, where we would like to tell you more about the hands-on things that we have covered so far in the genomics as well as in the transcriptomics part. Now I guess many of the SNU students are here, isn't it? So so the SNU students, Devalina, I know, Shomudi, Vaiman, thank you, Shubhriya, before you posted the link. Okay, so we don't have, thank you, Shomudi, for pasting the, for providing the, sir, I could not locate the log transform, I click on the PCR library and then end, it's all right. Yes, technically yes, but yes, Shubhrajit, thank you for, could you please explain the step before setting up the parameters, number of PC to 3? Okay, we'll cover that in the, please remember, I mean, we'll try to cover that in more detail when we go to the workshop next day. Yeah, Shomudi, we are from SNU, I know. So I think Devalina is also from SNU, and there are a few other people from SNU. Devanita, yes. Box plots, yes. I would tell you about the box plots. So, but you have to, you are not from SNU. Excellent. So don't worry. So this is meant for the SNU guys only. So maybe there will be an on-site workshop at SNU for the SNU people specifically. We have spoken to your administration and your controller of examination, Dr. Mala Mitra, or maybe your head of the department of your institute, Dr. Rajat Pal, is going to confirm us about the timing of it. But anyway, for the people who are attending online, the workshop will be happening on that day itself from 8 p.m. to 9 p.m. Okay, now going back, before I jump on into the question answer part, and we have this thing coming up. We have with us Mohit. Mohit is one of the faculties in this program. So if Mohit can elaborate a little bit on the program to other people, that would be very nice. Thank you very much, Mohit.
Thank you, Pratham. Yeah, I mean, there will be another free webinar on 10th of September. So this is a second free webinar for the part of the program that we are going to start, the Omics Logic Data Science program, which will be where we'll be talking more about the data science applications and machine learning applications. We'll understand more statistics, and we'll understand the different application of machine learning algorithms into the data science, into the data biomedical data. So those things will be taken into more detailed sessions. We'll have more detailed session, and it will run for few months with top international tutors. So that's what it is. It is going to start on 16th of September. So we are starting the Ilya Brodsky and Darko Medin will be taking the classes. So there is a pre-registration, so you can visit our website and you'll find the link, and you can see more about the program and understand what it is. And you can come back to us as well if you have any queries and if you are interested in joining the program. Thank you.
Yeah, thank you, Mohit. So one thing I would like to add, this will again be a Zoom meeting the way we do it in the SNU program or any other program across the globe. So all of you, I think, are now conversant enough to address, to use Zoom meeting. So that won't be a problem for you guys, I guess. Okay. Yes, Shubhrajit, thank you very much. For non-SNU participants, we'll definitely have, the session is on 9th. What is QN at level and quick QN at level 0 and QN? So how this affect the D? So that's what I showed you, Naresh. When you have quantile normalization at the level of 0, the significant signals are missing out there. So, and the significant gene expressions, and as well as the one that are really important for the next level of analysis are missing. So if you put a threshold value of 5, then it becomes more prominent. The things become more prominent. That's the sole reason of using the quantile normalization at the level of 5. 0 does not give you from sufficient information. Yeah, QN at level 0 will not give you. So log normalized with quantile normalized 5 at the threshold 5 will really remove all those noisy creatures from the data itself and will give you the fresh data that you can work on. That's what is the idea about quantile normalization. Okay, thank you, Naresh. Yes, thank you, Om Prakash, for today's. Yes, so all of you, I would request all of you to really go to Transcriptomics 2. So, and Transcriptomics 2 in detail, that contains very interesting features, and there are supplementary readings to it. So the broad overview that we have been giving by the virtue of these lectures will take you to the level. Actually, we are trying to point you to the the educational platform itself, and that will enable you to use the server as well and run around to demo pipelines to understand what kind of files can you expect, what kind of input can you give into the algorithms as well. Thereby, this will help you to do the project at your end when you are at the end of this course, or at the same time, it would help you people like Sunita. It will help people like Jyoti and Om Prakash and Naresh, who belong to the research fraternity and also the industry, to really use the platform in a proper way to come up with a plausible analysis and publish and also serve their respective clients. Yeah, Adebolina, you're from TAU. Yes, good to know that. Anything else? Any question? If you don't have any questions, we'd call it a day in that case.
Pratham sir, people are asking that who are not at or from SNU, how will they get the lecture? Will we be broadcasting it online?
Yeah, as I was telling you, that the people who are from Sister Nivedita University, specifically from Sister Nivedita University, you will probably have an on-site workshop on September 10th. But for the rest of the people who are joining from different parts of the globe or different parts of India, the timing of the workshop remains the same, September 10th, 8 p.m. to 9 p.m. So whether the SNU program happens or not, this program that we are going to organize on 10th from 8 p.m. to 9 p.m. remains constant. So don't worry about it. So so the SNU people are eager to host us in their in their institute to inculcate or to aware more students out there who have not joined probably. they might want to join, so that's why they want to make it free, but the decision is yet pending from their end. So we are that part is totally not yet decided, so none of you have to really worry about it. The SNU guys will know about it from their own administration, and for the rest of the folks across the globe. So yes, thank you, Prakash. Please complete the Transcriptomics 1, and now you have another thing on your plate, which is Transcriptomics 2. So that would be really nice, so that you can join with a proper understanding in the next class. So as I was telling you, 8 p.m. to 9 p.m. September 10th. That's the next day we see you again, all of you. Thank you very much.